Welcome to the Ongava Game Reserve. We're on a game drive and we've stopped at this beautiful clearing. I'm joined by Abby and we're going to just talk a little bit more about rhino conservation and also a bit about the genetics, lots of interesting stuff. And at the moment we've got Sharon. She's relaxed and grazing. And Abby, you know, this is a wonderful afternoon.
It is.
How are you doing?
Great, thanks. Can't be bad if there's a rhino behind you.
Awesome.
Yeah.
Yeah, so you're doing a little bit of genetics and working out some of the family trees of rhinos. How is that important in any way?
It's incredibly important. Everybody will be aware about the sort of array of threats that face rhino at the moment and, are threatening their survival. The most current and well-documented one at the moment is the threat of poaching.
Yeah.
And that's, decimating populations across the continent. But there are several other threats that are also equally impactful or have significant consequences for rhino populations. One of which is the sort of genetic aspect or genetic factors, particularly because of all the previous sort of impacts, anthropogenic impacts that have affected rhino populations. That causes significant issues with natural breeding systems and immigration. So the threat or the possibility of loss of genetic diversity could lead to inbreeding depression, which means ultimately a loss of fitness from the individual level to the population level for rhinos. And that could have significant consequences for their survival. So that's a significant factor that people should be looking at and contemplating when you're considering conservation management of rhino populations.
I think particularly when you have them in like a confined space, we've got
which
fence.
Pretty much all rhinos, except for a few core populations, are. Namibia is one of the few places that has some free roaming, literally free roaming rhinos. But basically, most of the other rhino populations you'll find are fenced in some way or another, or have geographical barriers which will affect breeding systems or mating systems within the populations. Ongava Game Reserve is one of those. I mean, we are a fenced reserve, a wildlife reserve, a sanctuary for rhinos. But it does mean that you have to have management programs in place for your wildlife populations, specifically endangered species populations or populations that have gone through genetic bottlenecks previously, such as rhino. So that it's something that you have to consider quite strongly, which we do here at Ongava. We have had... we have a very intensive rhino program. To date, we've actually been quite successful. We have both breeding populations of both black and white rhino, and we incorporate genetic management into our programs. So what we did was we established back in 2005 with Dr. Ken and Sabina Stratford, the directors of the research center. We started basically a parentage analysis of the white rhino population. We wanted to do that because we were at the stage where Ongava needed to sell or swap one of the dominant breeding bulls, and so we were actually looking at the genetic information in order to try and make the right decisions. It was meant to be an isolated project. We were going to look at the parentage analysis of white rhino and sort of tick it off and move very along. And the results that we got from the initial project were actually so impressive and led to just another array of questions and sort of threw into doubt traditional management techniques. And so we've actually continued with this project and we've expanded upon it, and we now run the project continuously, and we do it for the black rhino population as well.
Yeah. So on the game reserve, you own the white rhinos.
So white rhino are privately owned in Namibia, and then black rhino are you can be part of the custodian program through the Ministry of Environment and Tourism. And so the management techniques are slightly different in the sense that the white rhinos, which are privately owned, are generally typically more intensively managed, whereas the black rhino are not micromanaged to the same degree. So we don't remove the young bulls before they come into breeding age, which you do for white rhino. You don't typically swap out the dominant bulls. In the black rhino populations, they're sort of left, whereas in most white rhino populations, people will actively swap out their breeding bulls.
There was a time when a bull that was thought to have been the one that is less dominant swapped out, and in fact through your genetic work you found out that that was not the case, which it was quite interesting and really significant. What happened there?
Yeah, so when we did the initial parentage analysis, and we were actually able to assign parents for each individual offspring that we had, the results were actually quite impressive in the sense that they threw into doubt the traditional management techniques that I referred to. So typically the theory is that your territorial bull, white rhino bull, will be your main breeding bull. The results that we had when we actually did the genetic analysis was that that wasn't the case. So, for example, at that stage we were looking at, I think it was a cohort of 13 calves that could have been sired by 2 resident bulls. So we had 2 bulls, Bob and Derek, and we actually sold Bob because he was the dominant territorial bull. So he was always constantly marking, he had a very clearly, the largest territory, clearly demarcated, kept the females with him. And then the results actually showed that of the 13 calves we looked at, he had only sired 3 of them. So Derek had sired 10 of the 13 calves, and then traditional methods would have actually, kept him on.
Yeah.
So,
wow.
Yeah. so I mean, it has significant consequences for when you're selecting bulls to remove or to swap out. Just because behavioral observations alone are not sufficient.
And of course we were talking about how you collect the samples, and there are various methods, and you do it yourself on the game reserve?
Yes.
What's the process?
Yeah, so it's actually quite a lengthy process, and lately there's a few newer techniques that have come along which make it a little bit more affordable and more accessible, which is great for conservation genetics to go forwards. But at the stage when we're doing it, or we still do it, we started with non-invasive sampling. So we wanted to disturb the animals as little as possible. So we would try to collect dung samples. I did that for about 2 years and collected a significant amount of dung. For the black rhino, we found that the quality and quantity of DNA that we managed to extract was good enough to use, but for the white rhino it wasn't. So we had to switch to using tissue or blood samples for them. So we were able to biopsy dart some animals to get tissue samples from them. And then for the other animals, we actually we have an annual ear notching program, which may have been explained to you already previously, where we earmark all our rhinos to give them an individual identification. So during that time, every year, I can collect tissue samples or blood samples, which we use for the genetic analysis.
And then you do you do that in a lab, or?
Yeah, so actually then we set up an on-site small lab, so we do all our DNA extractions here on site. I do that, and then the actual processing of those samples, I take away just the DNA samples. And I used to do it in collaboration with the University of Cape Town, but I now do it at CCF, Cheetah Conservation Fund. They have a very advanced genetics lab there, so I go and process my samples there, which is very convenient, just down the road. I get to see some cheetah.
That's interesting.
So we do all the processing there, and then the actual analysis work is through software that we do, and then some manual analysis as well. Interestingly, with the white rhino, this was another thing that came up with the project, is that because their genetic variability is so low already, you actually can't just use the software to confidently assign parentage to all the offspring. So the software programs just can't differentiate enough, or to the degree that we wanted it to. So what we had to do is we had to overlay quite a lot of demographic data for the population. Now, we were really lucky in the sense that we have such an intensive rhino monitoring program on Ongava, so we actually had access, and that data was available. Most places won't have access to that, especially not over such a long time period. So that was particularly useful, and you do need that for the white rhino. Less so for the black rhino, but for the white rhino we had to overlay that in order to assign parentage for each individual to the degree of confidence that we wanted. Things that we discovered with the white rhino parentage analysis was that there seems to be, or appears to be, some form of mate selection occurring within our population. So females are definitely selecting for males that they are not related to, as opposed to related to, even if they have the choice. So we're overlaying that with a lot of territorial and home range behaviour to try and look at some possible mechanisms. Like I said, the mechanisms for mate selection are not known, but this could lead to, obviously, inbreeding avoidance, which is essential. So if that could be further understood, that would be critical for other populations as well, if that is the case. The other thing that we discovered was that the newly introduced bulls were actually breeding well before they'd established any home ranges or territories. So that also is quite significant for other populations that are bringing animals into theirs.
Yeah.
So there were a couple of very interesting and, as I said, impactful findings from that. And literally, the more that we look at, the more there's more questions than answers, really.
Wow.
Yeah.
That's amazing. The genetic bottling, is that how important is that for our country? And if we might lose them through poaching, and the safe havens would be one of the important areas that people travel to come and see these ancient creatures. That what you're doing here essentially is important.
Yes.
To make sure that you don't have that genetic variation or the species disappear.
Yeah, it's important on an individual level and on a population level. So obviously bigger is certainly better in this context. So larger populations are much more robust and are ideally what everybody should be aiming for. Unfortunately, sort of real life doesn't allow for that always. So as I mentioned, you end up with these small isolated populations. So in particular, the black rhino is a very interesting one. Some of the initial findings that we've had are very surprising and will have impact on conservation management, particularly on programs like the custodianship program, because I think what we'd like to do is expand that nationally, and we are in discussions with them to do that, to have a look at the meta populations and then look at the translocated populations, because what we've noticed with our population, the genetics support some of the theories that we've seen, which is basically we have these sort of two hotspots that have formed. So our population is kind of already split into two mini populations, if you like. And the genetics actually shows that they're not breeding between the two hotspots. So even on our small, property with a small population, you're kind of having separation within that already. So, I mean, generation after generation, that's going to have some impacts. So it would be fascinating to look at to see whether that's happening in other translocated properties, or populations, and also to see if that will have a larger impact, which it almost certainly will. And then to look at genetic variation within the different populations and to see if any diversity is being lost, if you're losing any alleles, just in all of the different hotspots, if that's even occurring somewhere else. I mean, we don't fully know why we have these two hotspots. Anecdotally, I think that happens elsewhere as well. And it would be fascinating to investigate, which we are, the mechanisms of why we have these two hotspots, because it's not geographically based. So something's causing them to kind of have these neighborhoods, if you like. But we don't quite yet fully understand why, and how that influences breeding structure and reproductive success within the populations.
Yeah. Oh, that's really, really interesting. And I think Sharon's talking to us.
So there you have two generations, right, walking past us. So that's also something that we'd like to look at, sort of, is there any loss over several generations? Is there any loss just in sort of the diversity of the markers that we're using? The nice thing, as I mentioned earlier, is that there are, DNA or genetic techniques are advancing from when we started out to what's available now. So you can look at quite a sort of diverse number of genetic questions, or at least address them, hopefully.
Yeah, no, that's impressive.
Yeah, it's pretty amazing.
Thank you so much, Abby, for sharing all of that information. And I'm really happy, and I'm sure all the viewers are, that what you are doing is incredibly important. And of course, like you said, not on the game reserve, but nationally. And of course, that will assure that we keep the variation, the genetic variation on a broader spectrum, and it will save the species.
It will certainly, hopefully, contribute towards it. It's one tool that must not be ignored in conservation management.
Yes.
It's not the only tool, and it also can't be taken out of context. But it's certainly critical with these endangered species, and is critical to their survival,
Awesome.
along with protecting them and everything else, and the habitat that they need.
Well, thank you so much. Everybody, that was an awesome afternoon. We thank Abby for what she's doing, and please do join us again for the next episode.